So far, we have concentrated on how to make DNA out of DNA. So this is very important for the application to pass the genetic information to the next progeny. From now on, we are going to concentrate on how the information that is stored in DNA is utilized in the cell. So you probably are all familiar with the central dogma. We can go from DNA to RNA, from RNA to protein, but we cannot come back. A very interesting part of life is that the DNA to RNA step is reversible. We indeed know many organisms who can actually make DNA out of RNA. So one very infamous example is the HIV virus. So we are going to get to that later. Or we even have organisms that make RNA from RNA. So we no longer can do this, but the infamous example of such an organism right now is the coronavirus. So now we are going to start looking at the transcription process, and we are going to start with the prokaryotes. In any given cell, we are going to have double-stranded DNA, and the information on this double-stranded DNA will be transcribed into a single-stranded RNA, and this language will then be translated into the protein language. But keep in mind, so on this slide, we only show RNA as a template for protein synthesis. As we will see later, RNA has many different functions in the cell than just coding for a protein. When we went through the chemistry of DNA, we also took along the RNA and saw that they are really very similar. So chemically speaking, we didn't spot that many differences. So one was on the sugar. The 2-carbon has in the RNA polymer an OH instead of an hydrogen atom. And the base thymine is a uracil in the RNA polymer. Again, the change is minor. There is a methyl group on the thymine, and uracil lacks this particular group. But when we now look at the structure of RNA and DNA, we see huge differences. Number one, DNA is a double helix, and this is it. But RNA is a very structurally diverse polymer, and we are going to see the multifaceted roles of RNA also in the next coming lectures. The chemistry of making a DNA polymer and an RNA polymer is the same. So in both cases, we are forming phosphodiester bonds. And the RNA polymerase also is unidirectional and transcribes from the 5' to 3' direction. These enzymes, although they seem to be very similar, they are indeed, evolutionary speaking, completely different proteins. And when we then go along and look at the RNA polymer formation and how RNA polymerase works, we are going to highlight the major differences between the DNA polymerase and RNA polymerase. So you remember Arthur Kornberg, who was the first to purify the DNA polymerase, and he also got the chemistry of DNA polymerization right. And for this, he was awarded the Nobel Prize in 1959. And guess who got awarded the Nobel Prize for the contributions to understand the molecular basis of transcription? Unbelievable but true. His son, Roger Kornberg, got the Nobel Prize. And when you then take into account that the other son from Arthur Kornberg, Thomas Kornberg, was the one who first purified the DNA polymerase 2 and 3. So the 3 is the polymerase that does the replication, as we learned in the previous lecture. Then we can say that this is probably unique that a family single-handedly contributes to a science like that. This is really very amazing. So let's take another look at the cellular architecture of a prokaryotic and the eukaryotic cell. We said that the major difference between these two is that the DNA is confined in the nucleus in a eukaryotic cell, and in the prokaryotic cell, we have the DNA exposed to the cytoplasm. And this architecture will now have consequences for the transcription. And we are going to start out with the easy transcription. And easy here is, of course, relative. In a prokaryotic cell, we have everything in place. So when the DNA gets transcribed into an RNA, this is then readily accessible to the translation machinery in order to make the protein. It looks very different in our cells. So the DNA in our cells is confined in the nucleus, and the transcription only occurs in the nucleus. And as opposed to the prokaryotic cell, when our RNA is made, that is not ready to go, it goes through a very elaborate processing. And once this is done, it needs to get out of the nucleus and go to the cytoplasm in order to be translated. So in the eukaryotes, the whole transcription machinery is separate from the translation machinery. The process that we are going to concentrate on is now circled here. We have the transcription that gives me an mRNA. So this is the so-called messenger RNA, and this type of RNA codes for a protein. And when I already make this distinction, then you can already imagine that we are going to have other types of RNA as well. On this slide, we see an overview of the transcription. And when you look at the steps, then this is more or less intuitive. Of course, you need to start somewhere, then make the polymer and end somewhere. Now, how easy that is. Now imagine you are the RNA polymerase and you have this circular genome of the E. coli with almost 5 million base pairs. And now you have to find your place, know exactly where to start and know exactly where to end. So much to easy transcription. Initiation. Where do I start? Remember the replication of the genome? How did the replication machinery know where exactly to start the replication? Well, the genome had a word for this particular machinery. It said, come here and start exactly here. So let's see if we can now find a word that the transcription machinery understands. If you will, this is a game of puzzle, just extreme. So what word does this transcription machinery understand in order to initiate the transcription? So in order to figure that out, people have aligned a bunch of genes and tried to see whether or not there is anything that resembles a common language in order to start the transcription. And indeed, they started to see the words of the transcription initiation. When you then write down the consensus sequence, then you really start to see the words. And here on the right, you see the so-called Tata box. And this is basically pretty much the same logic, having all these A's and T's lined up. The same logic that we have also encountered with the word origin of replication, where we also had this very conspicuous region of A's and T's. Why? Because we have two hydrogen bonds and they will tend to open up more easily to initiate, in this case, the transcription. Indeed, there are specified proteins that can recognize these Tata box, bind, and facilitate the opening up of the DNA in order to initiate the transcription. While we are at it, let's use the new vocabulary that we just learned. Remember the vector that we put together? We wanted to convince E. coli to also replicate our vector. For this, we put down the word origin of replication that the replication machinery will understand. And in order to make sure that the E. coli doesn't spit out our vector, we put in an antibiotic resistance. And this would be okay if we just wanted the E. coli to make more of this plasmid for us. But if we also want to continue E. coli or E. coli's machinery to do more than just the replication, then we can add our new word, promoter, meaning here is the word to start the transcription. With this promoter, we have another means of regulating the gene expression. Why? Because we can have very strong promoters in front of the gene, so the transcript is being made very fast. So I'm going to accumulate a lot of proteins in a short period of time, or I will have a weak promoter that, I don't know, makes couple transcripts every 10 minutes. That is going to be a complete different outcome. And this is basically the one of the means of the cell to regulate gene expression. And you can then understand that you really better do not get any mutations exactly in this promoter region. Now we know that the promoter is the word that the machinery understands, and this is where the transcription begins. Now, the big difference between the DNA replication and RNA replication. Remember, the replication machinery literally bends over backwards in order to synthesize both strands of the DNA at the same time in the same direction. Here, the RNA polymerase makes a single-stranded transcript, meaning it can encounter a gene on the top strand or the bottom strand. So in the polymerization language, it still goes from 5 to 3 prime, but both strands of the DNA can serve as a template for the transcript. Example, let's say I want to make a transcript of this coding strand, the black sequence. So this is where my gene is on, and I want a transcript of exactly that gene. So what will the RNA polymerase do? Remember, it has to synthesize 5 prime to 3 prime. So in order to recreate the black sequence, it will sit on the blue sequence, 3 to 5 prime, and start the synthesis 5 prime to 3 prime in order to recreate the exact same information. When you compare the pink and the black strands, you see that they are the same, except of course that we have a U instead of T. That's it. Before we go through the transcription step by step, let's take a look at the RNA polymerase machinery. As we have seen with the DNA polymerase machinery, we have many subunits. And as you can imagine, this is, of course, again, subject to research. So we are still learning more and more about these enzymes. But here you see an overview of the subunits. What we see here is the core enzyme. This actually needs another factor, the so-called sigma factor, in order to initiate the transcription process. And in the next couple of slides, we are going to go through how this transcription occurs step by step. So we now go back to our simple schematics and go through this transcription step by step. We have the word, we know the transcription initiation word, and the factor that can understand this word turns out to be this sigma factor, the glasses of the RNA polymerase, if you will, because you have to find your site among all these almost 5 million letters, right? And it turns out that there are different sigma factors that not only recognize the Tata consensus sequence, but other glasses that can read other words, if you will. So this is a more or less recurring concept. So we need to unwind the DNA again, of course, in order to get to the information. But the important information, the take-home message from this slide is that the RNA polymerase does not need any primer to start the transcription. This is a big difference to the DNA polymerase. Remember, when we talked about DNA polymerase, we talked about the universal common principles of all DNA polymerases. They all need a primer to start the synthesis. So this is the maybe one of the biggest differences between the DNA polymerase and RNA polymerase. When you boil down everything that we just talked about, then we can say, okay, we need the RNA polymerase. It needs to put on its glasses, find the word, and go. So is it that simple? Of course not. So we have our sigma factors, but the transcription involves also over 300 transcription factors. So the whole transcription business is really not straightforward, not even for a simple organism such as E. coli. Okay, now with the help of gazillion factors, we finally found our position with the help of the transcription factors, and we specified the promoter that we want to use, etc., etc. And now comes the elongation step. Once the RNA polymerase synthesizes a couple of nucleotides, it then breaks its contact with the promoter, and this then leads to a tight interaction between the RNA and the DNA template. Now we are at step 5. The sigma factor dissociates and the machine starts its synthesis, and then eventually you start the mRNA exiting the machinery and folding up into its secondary structure. So here again, differences between the RNA polymerase and DNA polymerase. So the velocity of the RNA polymerase is about 50 nucleotides per second. If you recall, the DNA polymerase goes at 1,000 nucleotides per second. But if you then think about it, it makes sense that the DNA polymerase is really fast because DNA polymerase has to do the whole genome, and mRNA transcript, I don't know, will be maybe a couple hundred. So the velocity may be indeed required in the case of the DNA polymerase, but not so much in the case of RNA. This seems to be another point where we have to ask ourselves whether or not this textbook knowledge that we went through is carved in stone. So what did we learn when we boiled down everything? So RNA polymerase needs its glasses in order to be able to read the word. But once the RNA polymerase finds its spot, it can just continue the elongation and the sigma factor leaves. Well, it doesn't seem to be that straightforward. This idea has been apparently challenged quite seriously. So if you are interested, you can go back to this overview, and there are indeed many different models that are apparently consistent with data. And it goes from the sigma factor really leaving completely up to sigma factor staying on top of the polymerase the whole time. So again, we have a nice example of how complicated life science is, and that we cannot take anything for granted, even if it is found in the textbook. Finally, we have the problem of the termination. Like initiation, how does the polymerase know where to stop exactly? And the question is now, what is the word for this one? It turns out that the termination is also non-trivial. So one simple example how it could work is depicted on the slide. So once the polymerase synthesizes a stretch that is palindromic and has many G's and C's, meaning it could form a hairpin. So it seems likely that the machinery would then stall after this. And the next bases that we see here are these repetitive U's, meaning these U's will pair with the A on the DNA strand, so the weakest of all combinations. So the RNA polymer would tend to release the DNA, and once the DNA reforms its double helix, the machine can then dissociate and we would then terminate the process. So in the next slide, I will show you a visualization of this transcription. So this is one particular movie that I like a lot because it also has more information how the gene regulation also works at this transcription level.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Why is it important to understand these specific steps of this machinery? Well, the same way with the replication machinery, if we know how the enzyme works, we can then start developing substances. Remember the E. coli that had invaded our intestine? The same way the replication machinery is a potent target, we can also attack the transcription machinery. You are surely aware of this problem of the multi-resistant bacteria. So the more we know, the better. So right now, the world has, of course, other problems with the coronavirus, but the World Health Organization actually thinks that the antibiotic resistance is one of the biggest threats to the human health. And if we do not find a way, we are going to probably run into serious problems in 30, 40 years from now. And when I say the humans develop substances like antibiotics against E. coli, etc., that we are not the ones who came up with this concept. We just found them. This concept of antibiotics goes back to the never-ending battle of giants, if you will, between bacteria, fungi, and mold, etc., early in the evolution. We are in the business only because we figured this out in 1928 that they make these toxic molecules to protect themselves. And thank God we figured it out early enough to save the lives of thousands of soldiers in the World War II who otherwise would have died from infected wounds. And for this, Fleming and others actually got the Nobel Prize right 1945, basically after the World War II ended. And when you look at the picture on the right, this penicillin, you actually are familiar with other types of this mold. So you may have not noticed, but you buy these molds from the supermarket. Penicillium camemberti or Penicillium roqueforti, and I'm really not making up these names. So those are the molds really of our daily lives. This was our journey to the easy transcription in prokaryotic cells. in the next lecture, we are going to take a look at the tricky transcription, how it works in our cells. And we are going to see that the concepts are the same, but we are going to have to deal with other problems. So as always, if you have any questions to part 6, please drop me a line.